Shall I start, sir?
Just wait. Madam, start.
Good morning to all the excellencies. Many of you made a huge effort to join us today. On behalf of all, we are deeply appreciative and offer you our most grateful welcome. We request all the participants to attend for the full scheduled webinar. You as participant can use the chat for questions to the speaker. E-certificate will be provided after submission of feedback. With these few remarks, I once again thank one and all for being a part of today's webinar. I, Dr. N. Lakshmi Sudipti, Faculty, Department of Pharmacology, feel privileged to extend a cheerful welcome to you all for this webinar on Ethical Principles of Laboratory Animal Experimentation in Biomedical Research, organized by Department of Pharmacology, KVSR Siddhartha College of Pharmaceutical Sciences, in association with APTI AP State Branch, sponsored by Siddhartha Academy of General and Technical Education. Now, let us have a look about our college profile. Coming to the theme of today's webinar, pharmacology is an essential component in the study of pharmacy and as well as biomedicine. Since experimental pharmacology involves the discovery of new drugs or to study the action of existing drugs and is done in two main stages. First stage, preclinical pharmacology, which involves the identification and optimization of novel chemical lead structure and testing on animals, animal tissues, or organs of their biological actions. The second stage, the clinical pharmacology, where the testing of drugs is done on human volunteers, patients. Experiments on animals are ethically acceptable if this has been demonstrated through the balancing of ethical concerns for each individual experiment. These include to enhance the life and health of human beings and animals, to study the structure, function, and behavior of living organisms, to educate and train in which no other possibility exists for achieving the necessary learning targets. The ethical approach of respect for life requires that the maximum gain in knowledge is achieved using the minimum possible number of laboratory animals and the limitation of their suffering to the essential minimum. Hence, today's webinar focuses on research tests on animals must conform to the established principles and precepts of science. Now, I request our adored principal, sir, Professor G. Devalrao Garu, to address the gathering.
Thank you, Dr. Sudipti. Good morning, one and all. I am very happy that our Department of Pharmacology, headed by Dr. Gnnavaram Kumar and his team, today they are organizing a webinar on ethical principles of laboratory animal experimentation in biomedical research. So this is a very useful topic. What I feel is whenever you want to go for what is known as clinical trials or clinical experimentation, animal models are very important, and animal experiment is also very important. And to explain this, what is known as various ethical principles of the laboratory animal experimentation in biomedical research, today with us we are having Professor Bharavi, Professor K. Bharavi from NTR College of Veterinary Sciences, Sri Venkateswara Veterinary University, Gnnavaram, and he is going to explain about what is known as all these ethical principles in a detailed manner. Now, my sincere appeal to all the staff and students from our college and other colleges also who are participating in this webinar, please make use of the points and important information going to be shared by Professor K. Bharavi sir. With these few remarks, I wish the webinar a grand success, and I wish all the participants also a good luck. Thank you. Thank you, Sudipti.
Now, I request our HOD sir, Dr. G. Vijay Kumar Garu, to give his message.
Thank you, Principal Sir, and thank you, Dr. Sudipti. Good morning, one and all. Myself, Dr. G. Vijay Kumar, on behalf of KVSR Siddhartha College of Pharmaceutical Sciences, welcomes you all for the webinar on ethical principles of laboratory animal experimentation in biomedical research, which is organized by the Department of Pharmacology in association with AP State Branch and ISFAR Vijayawada City Chapter. The use of animals in scientific research has been the area of concern in India. Given the sharp polarization of views between the animal welfare activities and the scientific community of the country regarding the use of animals. This led to the proliferation of litigation which impeded the pace of research. In order to eliminate the potential of conflict, in the year 2004, the Ministry of Environment and Forests set out to create a sound and cohesive regulatory framework for the use of animals in experimentation. Experiments should be designed with a minimum number of animals to give statistically valid result at 95% of level of confidence. The proper use of animals in experiments and avoidance of or minimization of pain and suffering inflicted on experimental animals should be an issue of priority for the research personnel, and unless the contrary is scientifically established, investigators should process on the basis that procedures that cause the pain or suffering in human beings will also cause similar pain or suffering in animals. The living condition of animals should be appropriate for their species and contribute their health and comfort. To enlighten further, we have eminent speaker, Dr. Bharavi Garu. We hope everyone will be benefited from this webinar. I'm looking forward for your patient listening and clarification. Wherever you need it during the webinar can be attended at the end of the session. I think it's going to be useful, good for the all faculty members and all the students who are attending this webinar. Thank you. Thank you, Sudip.
Now, the biodata of our today's resource person by Mr. P. Ravindra Babu garu.
Good morning, all of you. I am very happy to introduce, and it's great honor for me to introduce today's speaker, Dr. K. Bharavi garu, Professor, Department of Veterinary Pharmacology and Toxicology, NTR College of Veterinary Science, Gnnavaram, Sri Venkateswara Veterinary University. Sir completed BBSc and Animal Husbandry in the year 1992 and MBSc in the Department of Pharmacology and Toxicology in the year 1995 from the College of Veterinary Science, Rajendra Nagar, Hyderabad. Sir served as a veterinary assistant surgeon in Animal Husbandry Department, Government of Andhra Pradesh for 5 years. So Sir joined as the assistant professor in the Veterinary College in the year 1999 and completed his PhD in the year 2008 and became a professor in the year 2014. Sir guided 10 MBSc students as a chairman, published more than 25 papers in peer-reviewed national and international journals, and also presented research papers in various national and international conferences. Since 2010, he is involving in the activities of Institutional Animal Ethics Committee, and from 2013 onwards, acting as a chairman of Institutional Animal Ethical Committee of NTR College of Veterinary Science, Gannavaram. Sir is nominated CPCSA as a main link or other nominee to many educational institutions since 2010. And Sir attended training program for CPCSA nominee at National Institute of Animal Welfare, Haryana in the year of 2015. So Sir participated in the IEAC meetings as a chairman and as a nominee to many other institutions and scrutinized research protocols involving laboratory animals. Sir, we are very happy to have you here, sir. Now you can start, sir, your lecture.
Hello. Thank you, Ravindra Babu.
Thank you, sir.
Okay, today I'm going to talk on ethical principles of laboratory animal experimentation in biomedical research. So actually, these principles I compiled from the standard operating procedures of the CPCAC book, and also with the knowledge I gained after attending the CPCSE nominee at National Institute of Animal Welfare, Malabga. So during that training program, I visited National Institute Laboratory Animal Facilities at New Delhi. So with that knowledge, so I compiled these ethical principles of laboratory animal experimentation in biomedical research. So first, So animal research played a vital role in every medical advancement in the human health, human and animal health. And animals provide an invaluable and unique insights into the human system. So without animal models, so the study of human physiology or human the pharmacodynamics and pharmacokinetics of various drugs is it is almost impossible. And there is the indispensable need for animal research is recognized, and also it is supported by medical societies and health agencies around the world. In fact, every present day protocol of prevention, treatment, and cure and control of diseases and pain, suffering is based on the knowledge attained through the animal research only. And day by day, the new diseases are emerging. So we are all very well known the present day situation, the COVID-19, the pandemic is going like that. Day by day, new diseases may arrive. So, and there is a lot of biological science still to be studied. So, and future generations of humans and animals, they cannot be denied better health. And research involving animals is vital to continue medical progress in human and veterinary medicine. So this animal research has saved lives, saved this this with the help of animal research. So it is vital to continue medical progress in human and veterinary medicine. And animal research has saved lives, extended life expectancy, and improved the quality of life by enabling scientists to conduct critical experiments that identified ways to prevent, treat, and cure the disease. So ethically, this animal experimentation is defensible. Nowadays, many NGOs and animal ethics, the people who love animals, they protest against the using the animals for research experimentation. But in fact, ethically, animal experimentation is defensible, provided if you use responsibly, rationally, and humanely. Because experimentation procedures, the results, because this experimentation experimentation produces results that are in larger interest of the humanity and animals themselves. So first we are human beings, and of course then only we are scientists. So as a responsible human being, one should follow, promote, and verify the implementation of ethical practices in animal experimentations. And all personnel working in the laboratory animal must have deep ethical considerations. And it is important that such considerations are taken care at the individual level, at institute level, and finally at the national level. And using these animals in the research is a privilege granted by the society to the research community. Actually, because we are all answerable to the society, and because society ultimately we are all responsible, and whatever the research outcome it is coming, it is useful to the society only. So society expects that such use will provide a significant new knowledge or lead to improvement in the human and animal well-being. So for this, a set of ethical guidelines formulated by each country for implementation while animal experimentations. So which will reflect the concerns of the society and the scientific community at any given time. So this coming to the ethics in the animal use. So ethics, what is ethics? Ethics dealing with what is morally right and what is morally wrong. And also it is belief that something is very important. So especially the behavioral aspects of the human beings. So ethics evolved as a function of evolution of the concerns. So 100 years back, one practice is ethically correct, but nowadays, now it may be wrong. So it evolved with the time. So ethics evolved as a function of evolution of concerns and also as function of our developing understanding of animals as a sentient animals. That means they express their emotions, sentient animals. So therefore, implementation of ethical principles cannot be on fixed rules. So implementation of these ethics cannot be on fixed principles, but need to be evaluated as a function of increasing knowledge and technological progress. So day by day, the knowledge and technology increasing. So understanding of the animals is increasing. So one best example I will tell here. So animals, previously we keep all species of animals in a single room. But keeping the animals in different species in the single room will causes a lot of disturbance to the each species. So normally animal disturbs if the other species of animals present in the same room. So with that knowledge, so now we are keeping different species of animals in different rooms. So every country has provided a unified guidance to take care all such measures as may be necessary to ensure that all animals are not subjected to unnecessary pain and suffering before, during, and after performing the experiments at any stage. So before, during the growing period, or during the experimentation period, even after conducting of the experimentation also, animals should not be suffer with a pain or any other type of stress. So coming to the our country, that is government of India, considered a necessity of international norms regarding the ethical use of animals in biomedical research. So how to implement this ethical consideration? So to implement these ethical guidelines, a sound and cohesive framework to regulate animal use in experimentation was formulated. So for this, how these animal regulations were formed. So these regulations, so these persons or institutes engaged in scientific experiments on animals must act according to the provisions or rules of Prevention of Cruelty to Animals Act 1960. So actually, this act amended in 1960 itself, but ethical practices in animal experiments started during the 1998. So the rules the rule that is as per this Prevention of Cruelty to Animals Act, some rules are formulated. So those rules are breeding of and experiments on animal, that is control and supervision rules, 1988. So from 1988 onwards, the ethical principles, certain rules that are implemented during the animal experiments are started implementation. So to enforce these rules or provisions, a statutory body was created under a Ministry of Environment, Forest and Climate Change, that is CPCSA. So that is Committee for the Purpose of Control and Supervision of Experiments on Animals. So it is a statutory body under the Ministry of Environment, Forest and Climate Change, Government of India, and this body will implement, it will enforce the implementation of the previous rules, that is breeding of and experiments on animal control and supervision rules 1998. So this CPCSA, it is formulated certain guidelines. So first guidelines formulated and released in 2009. And these guidelines for experimentation on animals with animal welfare and well-being as its utmost concern. The most concern of these guidelines is the welfare and well-being of the animals. And there is a mandate for these guidelines. So mandate is of the CPCSE is to ensure that animals are not subjected to unnecessary pain or suffering before, during, or after performance of the experimentation. So there are certain goals for this CPCSA. So the goal is to promote the humane care of the animals and to provide information that will enhance the animal well-being and the quality of biomedical research and advancement of the biological or scientific knowledge that is relevant to the humans and animals. So these are all the goals of the CPCSA. So as per this CPCSA guidelines, every establishment has to is required to constitute institutional animal ethics committee and register with the CPCSA. And and also in addition to this, the institute also required to approve the animal house facility by the CPCSA and mandatory So every institute that is conducting experiments on animals must constitute animal ethics institutional animal ethics committee and also register with the CPCSC along with the approval of the facilities for conducting the animal experimentation. That may be either a building or if it is a large animal. So if it is a farm exclusively marked for the conducting the experiments. So to oversee the effective implementation of these guidelines at institute levels, and also to approve the project proposals, CPCSC nominates persons to every registered institute. And nominees, actually these nominees, they serve as a link between institute and CPCSA. They communicate, they regularly send the reports regarding the implementation of the animal ethical practices in conducting the animal experiments. So the key role of the nominee is to interpret and supervise and evaluation of the institutional animal care programs, and also to see that the CPCSC guidelines are implemented. And with the help of these CPCSC guidelines, the nominees scrutinize all projects and grant permission for conducting the projects on animals and evaluating the projects for their validity, whether the outcome is useful or not, whether the experiment they are conducting is in a proper way, ethical way, or scientifically it is valid or not. That also the ethical institutional animal ethics committee has to evaluate and also examine the rationale for implementation of the CPCSE guidelines. That is a 4 R's 4 R's concept. So here the 4 R that is nominees. First, we talk about the nominees. So here the CPCSE nominates 4 nominees. That is main nominee, link nominee, and the scientist from other institute and socially aware nominee. Whereas the institute nominates the chairman and animal house in charge as a member secretary, and scientists from 3 other disciplines. That is 3 scientists from the different disciplines. Total, the institutional animal ethics committee consists of 9 members. So to conduct the institutional animal ethics committee, the main nominee, chairman, and animal house in charge, these 3 nominees are must to conduct the institutional animal ethics committee meeting. And if main nominee expresses his inability to attend the meeting, then he can nominate the link nominee. So to conduct the meeting, 6 members quorum is a must. 6-member quorum is sufficient to conduct the animal ethics committee meeting. And the socially aware nominee. Socially aware nominee attendance is a must if the large animal protocols are scrutinized. So this institutional animal ethics committee will scrutinize the research protocols up to rodent species. That is up to rabbits. So in their phylogenetic order. So up to rabbits, they can scrutinize and give permission to conduct the animal experiments. Whereas beyond rabbits, that is on dogs, sheep, goat, cattle, and buffaloes, and the institutional animal ethics committee scrutinize and recommend the CPCSE for approval. So above rodents, the approval should be from the CPCSE. So these nominees are appointed by member secretary, secretary of the CPCSA, and these nominees will serve as the link between institute and CPCSE. And these one of the key function of this institutional animal ethics committee guidelines is to examine the rationale for implementation of the implementation of CPCSE guidelines, including the 4R principles. So the 3Rs, that is principle coined or concept mentioned by Russell and Butcher, that is reduction, refinement, and replacement. These 3R concepts, they are mentioned in their book, The Principles of Humane Experimentation Technique. This book published in 1959, and this is written by Russell and Butch. And the 3Rs concept are also included in the CPCSE ethical principles, along with the fourth R. The fourth R is included by the CPCSE, and that is rehabilitation. So now the CPCSE ethical principles includes the 4R concept, that is reduction, refinement, replacement, and rehabilitation of the animals. So this rehabilitation also a legal requirement in India, and states that the laboratory animals once used in experimentation must be rehabilitated. So this 4R concept is followed in the India. So coming to the ethical principles adopted by the CPCSA for use in animals in experimentation. So one by one, I will explain about the various ethical principles. So first, intended use. So all research communities, all research communities, they are answerable to the society, because we are all part of the society, which sustains on the society sustains on ethical values, and we are also use a public grant. So either maybe private or government institutes, ultimately we use the public grant to conduct these experiments. So we are all answerable to the society. So for approval and support animal experiments, public expects a clear understanding of expected benefits. So these benefits are advancement of a new discovery of physiological knowledge, or knowledge which is expected to be useful for saving or prolonging human life, or alleviating the suffering, or the significant gains in the well-being for the people of the country, or to combat any disease. So these outcomes, the people expects any one of these outcomes. So you use use suitable and intended use. No, so whenever conducting the research experiments on animals, use a suitable animal species for particular type of research. So, for example, there are different laboratory animal species are available. So, all the animals are not suitable for all types of research. So, if it is suitable, why so many species of animals? So, particular species is suitable to carry out research in particular types of research. So, for example, rats. Rats, we have Vister and Sprague-Dawley strains of the rats commonly used as laboratory animals. So rats are best suitable for toxicological screening, physiology and behavioral studies, and psychopharmacological studies, and gastric secretion, and ex vivo studies with isolated tissues or tissues of organs, and also infant drugs also best screened by using the rats. And psychopharmacological drugs and drugs acting on the CNS active drugs are also best screened by the these rats. And then comes to the mice. So commonly used one is Swiss albino mice, and it is a research model for the cancer, immune disorders, Alzheimer's disease, multiple sclerosis, transplant rejection. So all these types of research we can conduct best on the mice, and it is easy to produce transgenic mice like obese mice, newt mice, and dwarf mice. And also we can inactivate a specific gene of the mice. So we can produce a knockout mice by inactivating the various genes. So the mice with some deficiency in some enzyme or some hormone we can produce, and we can conduct the experiments on particular deficiency situation. And coming to the rabbits, so the commonly used is New Zealand white rabbit, and the rabbits are best suitable species for the cardiovascular system, skin to screen the various drugs for the skin sensitivity, and immune system disorders also the best. We can use rabbits. So to produce polyclonal antibodies. So the rabbits are used. These polyclonal antibodies are used in the vaccine manufacturing. So rabbit is the best species to produce the polyclonal antibodies, and bronchial asthma, stroke prevention treatments, and cystic fibrosis, cancers, pyrogen testing, skin and eye irritancy testing. For all these, rabbits are best suitable. Of course, and previously, the pyrogen testing of distilled water or intravenous fluids are carried out on the rabbits. So, of course, now this is replaced by the in vitro test. And coming to the guinea pigs, that is a Duncan Hartley guinea pig. It is commonly used. So guinea pigs are best suitable for a cardiovascular system and respiratory tract. So the respiratory tract of guinea pig is highest sensitive to the histamine. And toxicological studies involving the hypersensitive reaction can be studied best on the guinea pigs, and hearing experiments can be best conducted on guinea pigs. So guinea pigs have highest sensitive cochlear organism. Sorry, spelling wrong. Cochlear organism. Generally, all rodent species, that is mice, rats, guinea pig, rabbits, they have very good hearing sensitivity. They have highly sensitive cochlea. The hearing function is very well developed. Of course, they have a poor vision. So this guinea pig can be used to conduct any cochlear, the drugs which are toxic to the cochlear organ or hearing impairment. And coming to the infectious disease, particularly tuberculosis, can be best studied on guinea pigs, and bioassay of drugs like digitalis and also histamine we can conduct on guinea pigs, and ex vivo experiments with isolated organs, that is a tracheal ring, ileum, and all these things can be conducted on the guinea pigs. Then comes to the chicken. So chicken mainly used in veterinary medicine in applied studies, and embryonated eggs, they are best to culture the viruses. Virus frequently in veterinary colleges, virus are cultured into the embryonated eggs. So sterilely, virus is inoculated into the egg and incubated, and the virus multiplies in the egg, and the different viruses produce different types of lesions in the growing embryo. So basing on that lesion, so we can identify which virus is present in the or causing the disease. So then ileum and other organs collected from the slaughterhouse can be used to demonstrate UG practicals in the pharmacology department. So this can be ethically acceptable. We can collect from the slaughterhouse, so no objection will be there to use these isolated organs from the chicken to conduct. And in addition to this, there is zebrafish also. Nowadays, they are given. Zebrafish can be used for toxicological screening of various substances. And up to chicken only, the Institutional Animal Ethics Committee can scrutinize and approve protocols. And above the dog, we have to submit to the CPCS. So nowadays, instead of using dog, the mini pigs are commonly using especially in Europe and Western countries. The mini pigs are replaced in the dogs in conducting the animal experimentation because people have more attachment to the dogs, but they have less attachment to the pig. So people won't object to use pigs as experimental animals. And at the same time, at the end of experiment, we can sacrifice or slaughter the animal for table purpose also. So the people won't object much in using the mini pigs. Mini pigs are very small and you can manage very easily. Those are. So coming to the next principle, that is alternatives to the animals. So alternatives to use animals should be searched and should be prove their validity also. We have to search the alternatives as far as possible. We have to replace the animals. So for this, alternatives for animals should be searched and their validity of also should be proved. And while evaluating or testing the new products, as far as possible, you have to. And so coming to these alternatives, the cell cultures, tissue cultures, and organ cultures, and epidemiological studies. These through epidemiological studies, we can study the drug effects. So the best example is now COVID-19 infection. So COVID-19 infection, their pathogenesis, therapeutic protocols, everything studied on humans only. So no animal experiments are conducted. So the lot of the infectious is spreading like anything. So many people are affected, and the scientists got opportunity to study on the human beings itself. Actually, all therapeutic protocols are standardized on humans only. No animal experiments was conducted. Like that, the biotechnological research, various molecular docking methods, all these things can be used to study the drug effect. And like human volunteer studies and the computer and mathematical models are also developed to replace the animals in research. So this alternative should be searched and should prove their validity. And recently, there is certain agencies that is in United States under National Institute of Environmental and Health Sciences of US. There is one agency, that is Interagency Coordination Committee on Validation of Alternative Methods. So this agency ensures that new and revised alternative test methods are validated, and they develop, they approve the protocols that can be used as alternatives to animals in the biological research. And also they developed a computational approach to predict the human skin sensitization hazard without using the animals. So the computation, they feed all the data, and if you feed the molecular structure of any particular drug, then it will use the all information what we will get by conducting the animal experimentation. And there is another agency in Europe, that is European Union Reference Laboratory for alternatives to animal testing. So they developed and validated 5 different alternative tests, including the computer that is in silico model to study the the effects of various substances on a animal model. And also recently, organ on chips was developed. Here, it contains organ on chip contains human cells grown in a state of art system to mimic the structure and function of the human organs or organ system. So this is just like a big slide, the microscopic slide. In this slide, various channels are tunnels are drilled, and in these tunnels, the various organ cells are grown. So these chips are grown, and it will mimics as an organ system like liver chip and lung chip and kidney chip. All those are chips are available. So these can be used instead of animals in research, drug testing, and toxicity. And these chips replace human physiology, disease, and drug response more accurately than the crude animal experiments do. So instead of using the animals, and you can use the simple chip, and you can inject the drug, and you can see the what is the effect is producing on the particular organ. Suppose if it is liver chip, what is the effect is on the liver, and how the drug is metabolism. Like that, we can study. The results are more accurate than the live animal. Like that, the MATech epiderm. This is this is a three-dimensional human cell derived skin model. It replicates a normal skin. So by using this matrix epiderm, so we can determine skin allergic responses, evaluate chemicals for their ability to corrode and irritate the skin. So this matrix epiderm can replace the rabbits to test various the topically applied drugs on the on skin sensitivity, especially on the rabbits. And coming to the ethical, the third principle is a ethical source of animals. So acquire animals from a registered breeder only. So if animals are not available with the registered breeders, so then may be procured from alternate legal source with the prior permission from the competent authority. Suppose rats, mice, up to guinea pig, rabbits, we can there is registered breeders are there. We can procure. Suppose if you want to conduct experiments on sheep, goat, cattle, or buffalo, then the registered breeders are not available. Then we have to procure from the farmers. So then we have to take permission from your institutional animal ethics committee and procure from the alternative source. And one thing every researcher should keep in mind is that animals shall neither be killed nor removed from their natural habitat to carry out the experiment. This is as per the Wildlife Protection Act. We should not kill the animal or remove the animal from their natural habitat to carry our experimentation. So previously, we used to conduct experiments on the frogs and leeches they collected from their natural habitat. So the frogs rectus abdominis, the source of rectus abdominis muscle is used to conduct some in vitro experiments like that. So all such experiments are now banned. We should not collect the animals from their natural habitat. Then the fourth principle is degree of sentience. Degree of sentience. Sentience means responsive to. So responsive to the fear, stress, or response to the affection. So and all the animals, they are conscious of the sense impressions. They have certain impressions. So this the conscious is varies with the various animals. So depending on this degree of sentience, the animals are scaled with the different scaled basing on their degree of sentience in the phylogenetic scale. So if the alternatives are not available, then use animals with the lowest on their phylogenetic scale, that is least degree of sentience. That means responsiveness, the expressing their emotions to the fear or stress, all these things. Suppose these the phylogenetic order of these animals basing on degree of sentience is this is invertebrates. Invertebrates, that is cockroach and mosquitoes. So cockroach colonies and mosquito colonies we can use to estimate the pesticides, whether they are effective against these or not, and at how much concentration, at how much duration exposure they are killed. Like that we can estimate, and later on the same dose we can assess for their lethality in other species. Then comes to the fish. Fish also the less sentience, and birds. See, in invertebrate, cockroach, mosquitoes, and fishes, we cannot see any expression from them. For all type situations, they behave in the same manner. So there is no facial expressions. Of course, like that, birds also. If you give feed, or if you come close to the animals, also their behavior is same. So there is no change. There is no expression. So we can so use these animals and the rodents also. Rodents, mice, rats, and rabbits. They don't respond much. So if you give feed or if you pamper, they behave in the same manner. Whereas canines, felines, their behavior is different. If you with one second. Hello, am I audible?
Yes, sir, you're audible.
Okay. Okay, if alternatives are not available, then use animals with the lowest on their phylogenetic scale, that is least degree. So the lowest degree of sentience is cockroaches, mosquitoes, fish, birds, then rodents, then canines, felines, and bovines, equines, and primates, that is, and more evolved primates. So first preference is to invertebrates, cockroach and mosquito colonies, then fish. So fish can be used for toxicological studies, and zebrafish, especially the teratological effects of various drugs, can best screen on the zebrafish. Then birds, I already told, rodents, cat, like this. So anything higher than invertebrates in terms of level of sentience requires a regulation. Then comes to the welfare of the uh, experimental animals. So, welfare of experimental personnel using the experimental animals shall be responsible for the welfare of the animals, and this welfare denotes all the care provided for the animal, aiming at satisfying their physiological and behavioral needs. So their physiological and behavioral needs must be satisfied. So for this, animals should live in freedom for thirst and hunger and malnutrition. So to obtain their to satisfy their physiological and behavioral needs, animals must live in a condition that is freedom from thirst, hunger, and malnutrition. So for this, proper feeding and watering must be taken. So here, the feeding and water, we have to give sufficient feed only. So in the rat cage, that grill, normally we keep feed, no? So suppose a rat consumes 10 grams of the feed per day. Suppose 5 rats are in the cage. So you put 50 grams or 55 grams of the pelleted feed on the grill only. Don't put more feed, because if you put more feed, then different rats, there is a wastage of the feed, and also they will bite the feed and they will drink water, and the feed will be moistened. And after 1 day or 2 day, if you don't replace the feed, then there may be chance of fungus growth. And also the watering, you have to check every day and replace the water in the bottle every day. And you must take care of either the water leaking from the bottle, and if water is leaking, it will wet the bedding material, and it will cause disturbance to the animal. So that's why we have to take care feeding and water. So the feeding should not be more, and appropriate quantity of feed only we should put on the cages. And next, freedom from discomfort. They live with the freedom from discomfort. That is the proper ventilation, noise, and temperature. So animal houses are properly ventilated, and animal houses are airtight. Rooms should be airtight, and there should not be any windows, and all switches are outside the room only. And the each room should be pumped with air, that is filtered and cooled air into the room, and from the room the outlet, the air will go to the outside. The air should not recycle, so the only always the fresh filtered and cool air only should be pumped into the rooms. And as far as possible, there should not be any noise, so room should be airtight. So noise should not, outside noise should not come into the room. And temperature, we have to maintain 25 degrees of temperature throughout the experimental period. Then comes to the freedom from the pain, injury, disease. So in each case, they should not be overcrowded, and also all cages should be hygienically maintained. We have to clean the bedding material regularly. Daily, we have to clean the bedding material, and they should not be overcrowded, especially with regard to mice. Mice, they fight among themselves very frequently, and there is a cannibalism is there in the mice. So we have to, so we must see that overcrowding in mice cages should not be there. And mice has a habit of cannibalism. They bite other mice, and if any mice got injury and blood oozes, and it will appear as a red spot on the white hairy coat. No? So all other mice will go on bite that place, and that injury, that wound will become more and more day by day. And also some mice will acquire a taste to the blood, so they are go on bite, and they eat away also sometimes. So that's why, so if any mice is injured, we have to keep that mice in a separate cage. We should not mix with the other mice. And also we have to maintain the hygienic within the cage. And next, freedom to express normal behavior. So this is important. So for this, different species should be kept in a different rooms. So previously we used to keep different species of animals in the same room. So now it was found that the animals communicate with each other. So their sounds, we may not audible, the animals sounds are their communication system. So animals are disturbed, or they feel stress in the presence of other species of animals. So that's why the different species of animals should be kept in different rooms. Even their room doors also should not be opposite. The different species rooms doors should not be in opposite side also, because they emit some sounds, because animals are highly sensitive in hearing sense. So they communicate, and they cause some disturbance to the other species of animal, and thereby it will alters the various physiological parameters. And then comes to the freedom from fear and distress. Here, so animals normally they fear if the person comes and handle, and during handling, rough handling also, they feel distress. So for this, so every researcher, especially investigators, especially students who are working with the laboratory animals, the first time they must watch the animals. They must go to the animal house at least 4 to 5 times a day and interact with the animals. So while entering into the room, a laboratory animal house room, one has to enter with quietly, and while going, make some pleasant sounds, low and pleasant sounds. So the animal will recognize whenever you are coming, and then it will be friendly with you. We have to maintain some friendly emotion with the animal. So we may not observe those expressions in the rats and mice. It is very difficult to identify the emotions from the rats and mice. But we can observe when you handle the animals. So if you regularly visit the rat and mice houses, and by making small sounds or small whistle or saying something, then they will increase attachment with you. And whenever you are handling the animals to conduct the experiment or to give some oral feeding or to give injection, then they won't struggle much to your handling. Otherwise, if you go into the laboratory animals with a lot of sound, and you will visit the laboratory animals only once in 3 to 4 days, only you put the feed on the grill, a lot of feed, and put a big water bottle, and come once in 3 days by making a lot of noise. Then if you then you, while conducting experiment, if you handle, then rats will struggle like anything, and it will tries to bite you. And you while handling, you may lose the animal, and it will fall on the ground, and it will try to escape, and all these disturbance will create, that will create some alteration in the pharmacological parameters. That's why I advise to all the students, from the quarantine time onwards, you go to the laboratory animals at least 4 to 5 times a day, and make some pleasant sounds, and interact with them and spend with them. So then your experimentation will be very smooth without any trouble. And this is the model building floor plan of the small laboratory animal house for experimentation in educational this is for educational institute only, and a small house. So here you see this is the entrance. So at the entrance, one side office room and another side is storeroom. So office room is to keep all the registers and records relating to this animal use and animal house, whereas a storeroom to keep the feed and washed cages and all of whatever equipment we require in conducting experiments, we can keep in the storeroom. So this is sterilizing foot bath. So with this, we will enter from the main entrance into the room. So this laboratory animal house should have the hygienic corridor and a dirty corridor. So persons working with laboratory animals, they have to enter through this hygienic corridor into the room. So here for each species, there are 3 different rooms are there. This is the rat quarantine room. So here we will keep the rats after getting rats from any breeder or purchaser. First, we will keep the rats in the quarantine room, where rats are kept here for 1 week or more. And here all deworming, or if animal is sick, any antibiotics therapy or any B complex vitamins, all these gives to stabilize the rats or to adjust the rats to this environment. Once the rats are adjusted, then we will shift to the experimental room, and where the research experimentation is conducted in this lab. So here rats are divided into the groups and tagged, and they are kept for entire experimentation period here. And another one is a rat dosing room. So dosing room to administer any oral feeding, oral administration, or to give any injection. We should not perform those at the experimentation room. You bring the animals to the dosing room, and there you will give oral feeding or take weight of the rats, and again replace at the experimentation. So in the dosing room, there should be provisions for the dosing table, refrigerator, weighing balance, and all those things should be here. And coming like that for mice dosing. Here you see the doors of the each room, they are not opposite, and they are entirely on a different direction. And here mice quarantine room, mice experimentation room, like that for rabbit quarantine room, rabbit experimentation room, and rabbit dosing room. Like that we have to go. So once you enter into the room and do whatever work you want to do, then come out of this door into the dirty corridor. and even while changing the litter also, and take out all the litter and come out from the dirty corridor and exit through this way. And here there are two rooms are there. One is a washing and sterilization room. So to wash and the cages and other equipment and sterilization here, autoclaves and running water facility to clean and soak the cages, all those provisions will be there. And whereas here it is a dissection room. At the end of the experiment, if you want to sacrifice the animals, then all sacrifices we have to conduct here. And here we will conduct a dissection of the laboratory animal and collection of the organs or blood. All those things we will carry out here. And here the provisions for a dissection and a refrigerator, all those disposal of the carcass, all these should be here. And finally, exit is through this route. So, and if you want to again come to the office room and come through the passage from the outside only, you have to enter into the again room. So this is the model plan, and all these rooms are well ventilated. So they should be pumped, cooled, and filter air with a centralized air conditioning system. And out once it is pumped, the air should go out. So air should not be recycled, because here different species of animals in different rooms are there. No, if air mixes, so they sense the smell of other species and they cause a lot of disturbance to the animal also. So the air should not be mixed. So once the filtered and cooled air enter into the one room, that air should go out only. It should not be recycled. So this is the model plan of a small laboratory animal house for experimentation. And next principle is the number of animals in a study. Number of animals in a study. So this includes study has to be include appropriate number of animals necessary to reach a scientific objectives. So here the limit the number of animals used to a minimum necessary. See, here number should be minimum, and that also it is sufficient for a scientific validity of the study. So one should keep in mind that a study conducted with insufficient number of animals, then conclusion will be useless. If you in a objective of reducing number, if the number is insufficient to statistically to derive statistically significant value, then it is useless, and you have to repeat the experiment. And one should keep in mind, so the permissions will not be given to repeat the experimentation unless it is justified. Normally, once the experiments with the laboratory animals is started and you should finish successfully, there should not be failures in between, unless there is any unforced situations arrived. And conclusion of the study conducted with the insufficient number of animals is insufficient, and then scientists must justify number of animals to be used, and the committee also must review this justification. And scientists also they justify the number of animals, so and so number we are using, and committee also must review this justification to ensure that the appropriate number of animals are used. And here, the number of animals can best be estimated from a previous experience and by methods of resource equation and power analysis. So these are some of the statistical methods that is uh, evolved. So these are the some reference made articles. So the Festing et al. The article is the design of animal experiments, reducing the use of animal in research through better experimentation design. So this article is published by the London Royal Society of Medicine Press Limited, and another one is Ralph B. Del et al. And the sample size determination, that is published in Institute for Laboratory Animal Research Journal. So these two articles one can go through to find out the minimum number of animals in any study. So normally for routine studies for our drug evaluation in rats, mice, and guinea pigs, we usually keep a 10 number of animals per group. And we can keep, if rabbits are 6 number, that is sufficient. If we are conducting any new type of research, any specific research, then calculate the number of animals basing on these statistical procedures. So then comes to next principle, avoidance or minimization of pain and suffering. So, proper use of animals in experiments and avoidance or minimization. If minimization, when if avoidance is not possible of minimization of pain and suffering inflicted on animals, should be issue of priority for all the research personnel, and the pain infliction is scientifically, suppose if you are if you want to inflict the pain, so then it should be on the basis of that procedures or conditions that cause pain or suffering in human beings will also cause similar pain or suffering in animals. So this is, suppose as far as possible we have to avoid the pain or minimize the pain. So if the research demands, then only we can permit the induction of the pain. So the best example is the sciatic pain. Sciatic pain in humans, it causes a lot of problems. So for this, the CNS active drugs to suppress the pain and to regenerate the nervous system are used. So while evaluating such drugs, we have to create the sciatic pain situation in laboratory animals also. For this, the sciatic nerve is ligated and cause some damage to the nerve. Thereby, the animal also feels a sciatic pain. So here we are inflicting the pain. So here the pain induction is permitted because with the same conditions, we are going to evaluate the drugs for the same conditions in the human beings. In such a conditions only, the pain or suffering during the experiments are permitted. Then comes to the all scientific procedures adapted with animals that cause more than momentary or slight pain and our suffering should be performed with the appropriate sedation, analgesia, or anesthesia. So suppose if you want to give injection, no need of any anesthesia. Suppose if you want to give some implant or some suppose in castration or some minor surgical procedures, then use sedation, analgesia, and anesthesia. The best anesthesia that can be used in the laboratory animals, that is rat, mice, rabbit, is the ketamine. Of course, there are so many anesthetic agents they are telling. So the best one is ketamine. So it can induce immobility and sedation in the laboratory animals without causing any side effects. And this ketamine can be injected. So it is safe, non-irritant. So safe anesthetic for laboratory animals is ketamine. And handling in a careful manner is also important to minimize the pain infection, because one should practice the handling of animals. So different species of animals should be handled in different way. So you should handle in such a way that animal should not feel discomfort. If it feels discomfort, so it will struggle against your handling. It will tries to bind it, and you will throw the animal away. Then it will cause some injury or to the animal and creates a pain and stress. So to learn or to learn this handling, there is one certificate course in laboratory animal science. So this course is designed to promote humane, judicious, and scientific handling and management of lab animals. So this course is accredited by the FELSA, that is Federation of European Laboratory Animal Science Association, and this certificate course is organized by Madras Veterinary College, Vepari, Chennai, and they organize 2 to 3 times per year. And you go through the website of this Madras Veterinary College and register, and at least one faculty member from each college should attend this course, so that they can teach their students or whoever involving the animals in their research. Then comes to the next principle, that is post experimentation rehabilitation. So this is the fourth R, the introduced by the CPCSA. So here the investigators are responsible for the aftercare and or rehabilitation of the animals after experimentation. So cost of the aftercare or rehabilitation of the animals are to be part of the research cost. So initially the budget, while allotting budget, the rehabilitation cost also we have to include in the funding of the particular project. Sometimes animals are permitted to euthanize only in particular situations. So this is with regard to the large animals, that is the animals above a rabbit and above the species of the phylogenetic order. And the euthanize the animals when the animal is paralyzed or can no longer perceive the environment in a intelligible manner, and if animal left with severe recurring pain, and animal exhibits obvious signs of long-term extreme pain and suffering, or in situations where non-termination of the animal experiments upon would be life-threatening to the human beings or other animals. Suppose if you injected or if you created some infection which is highly pathogenic, then rehabilitating that animal is very dangerous, and it will spread the infection to other animals and human beings in such a condition. In these three situations, we can euthanize animal. And coming to the rat, mice, and the rabbit, at the end of the experiment, we'll sacrifice the animal, because most of our studies requires the pathological evaluation of a vital organ. So for that we have to collect the liver, kidney, and other vital organs. For that we need to sacrifice, and at the same time, rat, mice also we collect. Very often we collect the blood from the retrobulbar plexus. So there is some damage to the eye. So better to sacrifice the animal, so that we can study in detail the the effect of the drugs on vital organ functions. So while rehabilitating, the we have one has to follow the draft guidelines of the CPCSA for rehabilitation, reuse of large animals post experimentation. And in veterinary college, whenever we are conducting the experiments on the large animals like sheep, goat, or buffaloes, at the end of the experiment, these animals are not permitted to sacrifice. And at the end of the experiment, we will hand over to the college farm, where they are rehabilitated. They are mixed with the other farm animals, and they like that they lead their life. And majority of the times, we will not do experiments that cause locomotor disturbances or that cause permanent pain or such. Only the pharmacokinetic experiments are permitted in a large animals. And if a sheep and goat, if you conduct any experiment at the end of experiment, suppose if you want to study the effect of drugs on vital organ functions, those animals can be handed over to the that is a livestock production department, and they will slaughter for food purpose. And while slaughtering, then we can collect the vital organs, and we can study the effect of the drugs on the vital organ function also. Then the next principle is comfortable and healthy living conditions. Comfortable and healthy living conditions. So here, comfortable and healthy living, living conditions of the animals should be appropriate for the species and contribute to their health and comfort. Here, the veterinarians or other scientists trained and experienced in that proper care, handling, and use of the species will be persons responsible for housing, feeding, and caring. So, anyone, if you train any other scientist in as per that certificate course, they can be kept in charge, and they can train other persons also in taking care and handling and use of these species. So handling different species of live animal is entirely different. Different species handling is different. So we should not handle mice and rabbit in the same way. So while handling, it should not cause any injury. So especially while handling rabbits, don't lift the animal by holding the ears or skin. So that will causes a lot of damage and causes internal bleeding under the skin, and naturally it will disrupt the other parameters. And next, behavior is the focus of study of many experiments. And also it is a primary source of information about an animal's health and well-being. So behavior, we have to observe the behavior. So as I told you previously, at least 4 to 5 times you have to visit the laboratory animal house and spend the animal with them and observe their behavior. So in one see, we cannot observe the change in the behavior. So gradually we can identify the abnormal behavior of the animals. And these observation, we can go to the cages and we can observe. Sometimes if you reach the cages, so then animals keep quiet. So they won't express their abnormal behavior. So for that, every room of the laboratory animal house door, every the door of the laboratory animal house should provide a glass aperture to see through into the room. So from that room, if you watch the animals, then we can observe the abnormal behavior very in a better way. If you go to the animal, then they may stop their behavior like that. So we can observe from outside the room through the glass window, and we observe by with close observation after going to the cages also like that. We have to observe, and this behavioral change is the primary source of information about the animals health and well-being. So therefore investigators and their assistants be informed about the behavioral characteristics of their animal subjects. So the behavior of the rats is different to the rabbits, and the behavior of the guinea pigs is entirely different from the rats, mice, and rabbits. Like that, we have one must be aware of the animal specific behavior. So investigators must be aware of normal, species-specific behavior and unusual behavior that could forewarn the health problems of the animal. That is first initial sign of a health warning is the change in the behavior. So in conclusion, the the scientifically the proposal study using animals must be scientifically relevant and justify with the following objectives. So whatever proposal that is submitted to the institutional animal ethics committee, and it should be, we have to scrutinize that that proposal should be scientifically relevant and justify with these objectives. So the objectives are, so the proposal study should uh, uh, bring some outcome useful in the diagnosis, prevention, or treatment of disease and other defects in humans and animal or plants, and in evaluation of the activity, the efficacy and safety of medicine and for other biological and chemical substances and their composition, including radioactive elements or materials for therapeutic use in humans and animals, and also in evaluation of physiological parameters in humans and animals, and the quality control of foodstuffs. Also, we can use animals for in experiment. We can use the animals in experiments involving the quality control, and also basic and applied research, and also research for education purpose. In higher education institutes, the animal use is permitted, and also technical education and professional training leading to occupational activities involving experiments with animals, or in care and husbandry of laboratory animals, and also at finally the environmental protection, to protect the environment, to evaluate the effect of any agents that are released. The substances also can be tested on the laboratory animals. So coming to the responsibilities of the institutional animal ethics committee. So the institutional animal ethics committee strictly follow the CPSC guidelines and abide these by the CPSC rules. So the institute should maintain proper records and made available to the nominee or any person authorized by CPSC at the time of infection. and they strictly follow the CPSC guidelines while procuring, stocking, and rehabilitation of the animal. These are the major responsibilities of the CPSC. And coming to the records to be maintained by the Institutional Animal Ethics Committee, that is the record of registration of institute and animal house facility approval with the CPCSA. So we have to renew this. Initially, you have to register and renew this registration at every 5 years. And there is minutes book of the Institutional Animal Ethics Committee meeting. So in meeting, all the discussions should be recorded, and a Xerox copy of this the minutes should be uploaded into the CPCSC website by the main nominee. And next comes to the Form B file. It is a file, so it is nothing but a Form B is nothing but application for a permission for animal experiment. So this application is submitted by each investigator to the Institutional Animal Ethics Committee. In duplicate, they have to submit. So after evaluation, the signed and approved copy, one copy will be given to the investigator, and one copy should be kept with the office in a file, Form B file. And then comes to Form D register. So this is the record of animals acquired and experiments performed. So all the animals that are entering into the laboratory animal after purchasing should be entered into this register. And how many days you are keeping in the laboratory animal. And at the end of experiment, whether the animals are sacrificed or they are rehabilitated, every detail should be in the Form D register. And this Form D register should be in the office of the laboratory animal house. So in the front, the office room is there, no, in laboratory animal house. In that house, this register should be kept. And also visitors register. So any visitor, either CPCSC nominee or any person that is endorsed by the CPCSC, when visits the laboratory animal house, they must sign on this register. So the visitors register also made available at the laboratory animal house. So in addition to the ethics committee is not a scientific committee. So it is strictly speaking, it only scrutinize any ethical issues are implementing or not. We are not ethical committee should not interfere in scientific aspects. So ethical evaluation should include evaluation of expected benefit. So the relevance of animal model with regard to the subject objectives, whether the objectives with this animal model are achieved or not. That only we have to say. So in what way or how which species they selected, that is immaterial for us. Whether that species selected is suitable, and with that species expected benefit is obtained or not, that only we have to see. And the possibility of replacement. So if any possibility for the replacement is available, so need to use animals to reach the study and objective. So that is if possibilities alternatives are there, we have to suggest those alternatives. And if alternatives are not available, then we can use these animals to study the use animals to obtain that study objective. And then possibility of refinement and improvement. So the suffering and distress to the animals, as well as the procedures implemented to minimize these suffering also we have to advise. So as far as possible, we have to monitor. Suppose any painful procedures are there, or any implants are is kept in the animal's body, then they have to mention all those procedures in detail, and how they are conducting, how they are eliminating pain and suffering during those procedures also they have to mention. And if satisfied, then only the ethics committee will give permission. And likelihood of reaching this objective by the methods proposed. So we are not involving in the technological issue, but we see that the methods proposed are suitable to achieve the objectives of the study or not. Either we have to see. That should be suppose if by giving some drug, there is oxidative stress is creating. So for that, what are the oxidative parameters? All should be estimated. So leaving the oxidative parameters, if you estimate some other parameters, blood picture, all those things, and without estimating the peroxidative markers or oxidative markers, it is useless. So like that, we have to see that the methodology also we have to check. Then the techniques and the methods to be used. So based on techniques also, whether they are relevant or methods are relevant or not, we have to say that is institutional animal ethics committee should scrutinize. So basing on these elements, the committee will be able to give an opinion on the study, which should be given before the beginning of the study. So based on the permission only, the experiment should be started. If they are rejected, the experiment should not be conducted. For the species up to rodents. So above the species, they are scrutinized and recommend to the CPSAC for giving the permission. Once CPSAC gave permission, then they can start experiments on animals above the rabbit in their phylogenetic order, like rats, dogs, pigs, sheep and goat, and cattle. So coming to the retrospective assessment, once the permission is given, the institutional animal ethics committee will also retrospectively study the experiment that is conducted. So here, the examining the development and results of the study. So this study, a retrospective study, particularly recommended for studies which are new to the committee, or for long-lasting programs or studies which are potentially stressful or painful to the animals. In such studies, either long-lasting programs means the studies for 3 months, studies carried for more than 3 months or 9 months, like that. In such a case, for chronic toxicity conditions, or if animals are exposing to the stressful or painful situations, then also we have to retrospectively assess the laboratory animal experimentation. And so that is with this according to these principles, one should carry out their experiment by minimizing the pain, suffering, and of the animals. Of course, this is the responsibility of the every citizen, and every society expects the ethical practices while conducting the animal experiments. So thank you for listening patiently.
Thank you, sir. Thank you, sir, for your nice explanation, sir. Sir, myself, A.V.S. Ravisainath, Assistant Professor, Department of Pharmacology, on behalf of KV Sir Siddhartha College of Pharmaceutical Sciences, take my privilege to propose vote of thanks on this knowledge pondering webinar, Ethical Principles of Laboratory Animal Experimentation in Biomedical Research. Thank you, sir, for providing deep insights into the ethical considerations in laboratory animal experimentation of biomedical research. Sir, we are very thankful to you, sir, for delivering the role of various committees and the organizations in the experimental protocols of laboratory animals. Thank you, sir, for enlightening us with the concepts of biomedical research, sir. Thank you very much, sir. And I thank our beloved principal, Dr. G. Devalrao Garu, head Dr. G. Vijay Kumar Garu, colleagues and management for giving me this valuable opportunity. Last but not the least, I thank all the audience for their patience listening and making this webinar a grand success. Thank you, one and all.
Thank you. Thank you.